Well, good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Effective Study Design and Execution of Renal and Hepatic Impairment Trials. My name is Ryan Muse, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the Comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available to those who registered on xtalks.com. At this point, I'd like to thank Dr. Vince Clinical Research, who developed the content for this presentation. Dr. Vince Clinical Research, DVCR, is a full-service contract research organization specializing in early-phase trials in both healthy, normal volunteers and patient populations across a wide range of trial designs and therapeutic areas such as neuroscience, substance abuse, pain, cardiometabolic disorders, infectious diseases, and many others. CRO services include project management, data management, biostatistics, statistical programming, PK/PD analysis, medical writing, monitoring, as well as site feasibility and management for multi-site trials. And now, without further ado, I'd like to hand the mic over to Dr. Brad Vince.
Thank you, Ryan, and good afternoon, everyone. My name is Brad Vince, and I'm the CEO and Chief Medical Officer of Dr. Vince Clinical Research. I want to thank you all for joining our webinar today on the effective study design and execution of renal and hepatic clinical trials. Most importantly, I would like to personally thank our 4 esteemed speakers for sharing their expertise on these challenging studies. Now, these trials must often be conducted in order to assess organ impact on PK parameters of your investigational product. We have several speakers today that will be providing their insight and expertise in the conduct of these trials, which we'll hope you'll find helpful. Now, getting a subject matter expert involved early, such as Dr. Harry Alcorn, is critical, not just for study design, but also for inclusion-exclusion criteria. It's critical in ensuring that these CSRs are delivered on time and as promised. And recruitment is traditionally one of the rate-limiting steps to the successful execution of these studies. We're also going to be discussing population PK modeling in combination with adaptive trial design. This in order to potentially reduce the number of enrolled subjects in these clinical trials and also potentially save time and money. So thank you again for joining today. And now back to you, Ryan.
Thank you, Dr. Vince. Now I'd like to introduce our first speaker for today's event. Corey Nevitt, Executive Director of Site Partnerships, has been in clinical research for over 2 decades with robust experience in operations, commercial strategy and planning, and global site partnerships. Corey is responsible for the growth and expansion of strategically aligned site partners and ensuring that the multi-site studies are conducted by the most capable investigators.
Thank you, Ryan and X-Talks for this opportunity. And thank you for all of you joining us today. This could be a 2-day workshop on this topic alone, but in the essence of time, I'm going to focus on these key areas to successfully managing impairment trials. It starts with site alignment. We'll talk about the unicorns, aligning with those cornerstone sites, and essentially innovating the basics. We got to enhance efficiencies. How do we truly reduce site and sponsor burden? And aligning and then realigning expectations through the life cycle of the study. This is needed early and often. From design changes to study delays, we have to have the ability to adapt and change quickly. Removing silos is easier said than done, I realize that, but we need to consolidate function and task where we can. And then there's transparency. Through the good, the bad, and the ugly, open and honest dialogue obviously is key. Next slide, please. So what are the current challenges? First is site demand. We all know there are a limited number of unicorns out there, so we need to adapt and innovate. Timelines. How urgent is it? What are the critical milestones? Is it to inform the dosage for your later phase study? Is it for an NDA, a PMR? Early site engagement is critical at this stage. Assessing competing work. Each protocol is unique, and so are the patients. Holistically, competing work can be a positive, in my opinion. The patients can enroll in that study that fits their criteria, which ultimately encourages future participation. Study designs. We have to get site feedback early on. It's so critical. This includes discussions around IE, comorbidities, con meds, and Harry will be touching on this in a few slides in greater detail. Lastly, we got to avoid the telephone game when it comes to communication. Siloed groups is a recipe for disaster. Next slide, please. So here are some key considerations for the CROs. When it comes to quality versus quantity, it shouldn't be a numbers game. You got to partner and invest in those sites that align with your model. And when it comes to trust, we all know relationships matter. Trust the feasibility, trust the budgets, trust the feedback, trust the recommendations. Having humility is a strength. Know what you don't know. Welcome the site feedback with open arms. Issues will arise, but we need to be proactive and not reactive. An appropriate escalation pathway with triggers should mitigate the number of surprises, if you will, down the road. Lastly, delays in site payments. It impacts overall site engagement, study priority, allocation of resources, and data quality. Next slide, please. So what is a site-centric model? Well, in a nutshell, it's putting the site first, trusting the relationship, ensuring timely and quality deliverables for our studies. You got to centralize the functions, consolidate the tasks, remove those silos, streamline everything from inquiry all the way through site activation. Then you have the site ID and feasibility. You got to take a proactive approach to essential doc collection. You got to eliminate the redundancy and reduce the site burden and sponsor burden. And when it comes to budgeting contracts, negotiate those rates up front. We need flexible terms so site gets paid faster. And we have to avoid unnecessary and costly change orders. Again, this escalation pathway is critical. This is a single point of contact that can support and guide when needed. Next slide, please. Some additional considerations for success. From PM through support services, the project team needs all these qualities below. You're only as good as your weakest link. You got to align with an IRB that understands early phase patient studies. Specifically when it comes to IE criteria, you got to ensure the review cycles are timely and that they can provide rapid responses. And lastly, you got to proactively identify the support that is needed up front. You got to push for additional funds to enhance recruitment. Ensure translational services are scoped out to avoid those delays, and have resources in place for local lab support and the DTAs. What lab supplies are needed versus a central lab? How can you support the sites? Lastly, ensure appropriate processes are in place to avoid dual enrollment for not only patient safety, but also data integrity. So how do we grow? We have to change the status quo. We have to be transparent with all parties. We have to align with our sites, and specifically those sites that emulate your organization. So in the spirit of humility, I'm going to pass the baton off to the expert, my friend, longtime colleague, and trusted mentor, Dr. Harry Alcorn.
Thank you, Corey. Now I'd like to introduce our next speaker. Dr. Harry Alcorn, PharmD, is Chief Scientific Officer, Phase 1 Site Tampa at Alexia, and has over 30 years of clinical research working with public and private biotech and pharmaceutical companies in studies of kidney diseases, diabetic, hepatic, and cardiovascular patients. He has designed and authored protocols and has been consultant to a multitude of companies in the industry on protocol development, clinical execution in specialty populations, and regulatory guidance.
Thank you, Corey, Dr. Vince, and Ryan. It's my pleasure to have this opportunity to share my experience in these specialty population studies. It's my passion for the last 20 years to conduct renal and hepatic studies, where I've been PI on over 70 renal studies, sub-I on over 100 studies, and written over 50 renal protocols and presented to the FDA 5 times. Let me first start with the renal studies and then address the hepatic studies towards the end. Next slide. Specialty populations. Renal patients, who are they and where are they? CKD patients, mild and moderate, are unique. They typically don't even know they have reduced kidney function. They may or not have symptoms. And on multiple medications possibly. And on these medications, can they be DC'd or even changed? 80% of these patients with renal impairment are diabetic and or hypertensive. Hemodialysis and peritoneal dialysis patients are easier to identify as they have a weekly schedule for dialysis. You actually know where they are and how best to contact them, and they're usually most favorable to considering research. Next slide. As Corey mentioned, as well as Dr. Vince, recruitment is probably the biggest challenge with any clinical trial, and within specialty populations, it is even more difficult. As with any clinical study recruitment, the number one issue that impacts timelines and costs greatly is this recruitment effort. It is critical the site has access to these patients. Alexia is positioned uniquely in that we're contractually aligned with the physicians and their clinics for direct access of their EMR, Epic, to actually do data mining for the inclusion-exclusion criteria for targeted recruitment, along with physician support once identified. To complement this, staff knowledge of the disease that causes reduced renal function is also effective in our communication for recruitment, understanding their comorbidities, symptoms that may be considered normal, being at their age that they are older, what medications they may be on, and diets respectively. Next slide, please. Again, complementing Corey's position on protocol design being extremely critical, which it is. Having the CRO, the site, and the sponsor engaged from day one has produced the best of protocols in meeting sponsors' endpoints with realistic inclusion-exclusion criteria for timely, accurate, and complete data. The FDA offers options here with the renal studies, and specifically a guidance. The full study guidance, which identifies mild, moderate, severes, hemodialysis or peritoneal dialysis patients, and then identifying those one-to-one by age, sex, and BMI for a full study design. There is an option here with the matching criteria in pooling your renal groups and then averaging the data for matching purposes. The difference is that you must enroll all the renal subjects first to be able to average the data for matching. Timelines and costs need to be considered. Reduced studies are available and identified by the FDA for drugs that are predominantly eliminated via non-renal routes. Next slide, please. Again, understanding that specialty populations are not the same as normal patients, and within protocol design, central labs, values, one must be thoughtful and complete. Specialty population labs will differ from normals, and not to compound extra work for the site, the CSR, or for the sponsor, the central lab will need to have normal ranges for this population to avoid the non-clinically significant results. IRB knowledge of specific populations also is a critical role. If they're not familiar with specialty populations, they will push back with questions and recommendations that are not realistic, which also leads to delays in starting the study by weeks. For example, laboratory values identified above, but specifically QTC and blood pressure. Urine collection for the protocol design may be overlooked. These time points need to be addressed as not to interfere with IP administration, and that the staff has the knowledge and experience actually how to carry this out logistically if they are pooling the urine or if there's any special processing required. For the site to have specific bathrooms that can be locked to assure that participant voids can be collected and there will be none missed. Next slide, please. Hepatic studies, not unlike renal studies, have similar considerations, understanding being different disease state, and to that point, what scoring will be used. Typically, it's Child-Pugh. The staff, again, should have knowledge of the disease state with liver impairment for effective communication with patient recruitment, understanding that these patients usually have greater alcohol consumption, hepatic infections, IV drug use, obesity, or even family heredity issues that need to be part of the conversation to ensure effective recruitment. Next slide, please. Renal and hepatic patients are at best hard to find, and I wanted to provide an overview of what has made our studies successful in the past. And so I've called out the key criteria within the inclusion criteria, exclusion criteria that have truly been the sticking points as it relates to recruitment and enrollment for our studies. The age, increasing it to 80 if possible, as we all know 75-year-olds are not all the same. The BMI, increasing this to 42, as these disease states are most commonly associated with larger BMIs. QTC guidelines, up to 470 for males and 480s for females. And the urine drug screens, be careful what you ask for and be clear on potential drug-drug interactions. Limit your drugs to those that have direct DDI and possible limiting the CYP to strong and moderate and allow for substrates. Be careful what you ask for and be clear on potential DDIs. Limit your drugs to those that have a direct drug-drug interaction, and if possible, put a limitation on your CYP enzymes to strongs and moderates and allow for the substrates. As it relates to other drugs that are tested on UDS, TTAs have multiple false positives for other drugs. And with TCAs in this arena, you'll have false positives that potentially could rule your participants out of the study. This population is very prone to tobacco and marijuana use, and you will see this as a high usage in this population and will have a huge impact on recruitment if not allowed. This is a team that knows how to work together to get these studies done right. The CRO having the right staff, knowledge, and experience. The site having access to patient population, ability to mine data and find patients and reduce recruitment timelines and cost. Staff knowledge, experience with specialty populations. And let me be a little more specific here. In assuring the participant safety, attention to the details with procedures, symptoms, medications to reduce AEs. Offering changes to the primary care physician in current medications for ones that would be allowed by the protocol. And specifically in the dialysis population, dialysis collection requires special training and very early hours of the day to be on board. Dialysis filter consideration in the protocol needs to be understood, if it should be standardized, and when would IP dosing occur, and IP dosing pre or post dialysis session, you need to understand the half-life of the drug. And if you want to maximize the PK time points between your dosing and the next dialysis session, you will need to dose on the third dialysis session of the week to allow for 2 days to the next dialysis section to maximize your PK time points. These details are critical for obtaining the quality data and the specialty population specifically. But it's only through the relationships that we've had with Dr. Vince and his team that we've been successful in continuing these type of specialty studies. And these are ones that are difficult at best, as I defined, but ones that, if the staff and the clinical group are understanding of what is needed, they are easily conducted and timelines are met.
Thank you, Dr. Alcorn. Now I'd like to introduce our next speaker. Dr. George Ati, MD, is a principal investigator with over 35 years of experience in clinical research and has participated as an investigator in well over 500 clinical studies. Before his role with Dr. Vince Clinical Research, Dr. Ati served in various leadership roles such as Vice President, Medical Director, Site Director, and Lab Director for multiple CROs.
Thank you, Ryan, for the introduction. My name is George Ati. As per my intro, I've been doing this for quite some time. I've been doing phase 1 for coming up on 20 years now, and have done multiple renal and hepatic trials. What I'd like to go over is some of the common abnormal labs that are encountered with this population, with these populations. Next slide, please. So let's start with renal insufficiency. As you all know or are familiar with, there's multiple stages of renal insufficiency. Anything between 60 and 89 is usually considered mild, and between 30 and 60 is considered moderate. As recently, this has been modified somewhat and sort of subcategorized. But it's pretty straightforward when it comes to obtaining these values. You can see there at that last bullet is the CKD EPI creatinine equation. This was updated in 2021, and it definitely will have a significant impact on your recruitment and finding subjects. Next slide, please. So I'm going to go ahead and start with the eGFR calculation. As you can see, it's fairly complex, and one of the things that has changed since 2021 is previously race was included as a variable. It's no longer included as a variable. So basically, you have age, sex, and creatinine. When you're dealing with this population, you're going to frequently encounter other abnormalities. One of the most common one will be hemoglobin. As you can imagine, with decreased renal function, there'll also be a tendency towards anemia. This usually is not a major issue. It, in fact, is rarely seen unless you get into the fairly significant levels of CKD. Now, just as an example, with regards to creatinine, minor little changes make a big difference. On the last 2 bullets, you'll see the same creatinine based on 2 different individuals, a 21-year-old male and then a 57-year-old female. You can see that the 21-year-old male has a normal eGFR of 98, whereas the female with this exact same creatinine would be considered in the moderate range. This is something to consider when you're looking through your database or your practice. Again, very minor changes or small changes can create a possibility for enrollment. So if you're not sure, one of the easiest things to do is to actually run some calculations, check your database, check the last creatinines, and see where they fall. Because when you're doing these types of trials, it's important, obviously, in most cases, to have mild, moderates, and severes. You're probably not going to be handling those moderates and/or severes in your practice, but those milds are out there, especially with advancing age. Next slide, please. Okay. When you're dealing with patients with decreased renal function, the 2 of the common abnormalities are hyperkalemia and hyperphosphatemia. Again, increasing degrees of CKD make these much more common. However, it's important to understand some of the drivers for that. For hyperkalemia, treatment with ACEs and ARBs definitely can increase the potassium levels. If they have some underlying heart failure, decreased perfusion adds to that. And of course, just overall decrease in renin activity is a contributor. So the potassium intake is the major controlling factor in this regard. Again, with also with hyperphosphatemia, you're seeing the same types of situations where patients are more likely to have that. And again, with increasing levels of failure, you will see these things become more apparent. Now, both of those are important because both can be controlled with diet. So if you have patients and they are in an inpatient setting, your meal prep for those particular patients is critical. You definitely don't want to overload them with foods that are high in potassium or phosphorus. However, if they are on binders or they are taking drugs to help counteract the underlying abnormalities, then you also have to be careful there to monitor carefully. Next slide, please. As Dr. Alcorn mentioned, the Child-Pugh scoring system is used for determining the level of hepatic insufficiency. As you can see on the graph on the table, bilirubin, albumin, prothrombin times, and/or INRs are the laboratory values that will be critical to determine whether or not the Child-Pugh score is appropriate. The other findings, encephalopathy and ascites, are less precise, but generally are easy enough to accommodate, and obviously encephalopathy is very apparent when it's present, so that usually is not a problem. Next slide. So the other labs that will be frequently found to be abnormal are those you would expect. ALT, AST, GGT, the alk phos, and platelets all can be out of range, sometimes significantly out of range. And just like the situation in CKD with hemoglobin and potassium and phosphorus, you will commonly see alterations and aberrations that you're not used to seeing in regular patients. And that's one of the issues with regard to these types of studies. If you're a PI on these studies and you're not used to this population, it can be a little bit intimidating. The good news is that very infrequently is there anything required to modify these results. The patients are coming in, they have underlying disease, they have known disease, their baselines are established. So even though you're seeing some significant abnormalities that would generally potentially trigger a referral to a specialist, you have to understand that this is their baseline, this is where they live, and it's just a matter of monitoring to make sure there aren't any dramatic changes, and giving some thought to diet and of course all the other medications that they may or may not be on. Now I'd like to turn it over to Dr. Tony Grant.
Thank you, Dr. Atee. Now I'd like to introduce our next speaker for today. Dr. Tony Grant, PhD, is the Associate Director of Pharmacokinetics at Dr. Vince Clinical Research. In her over 17 years in the industry, she has worked across all study phases and has extensive experience in early phase clinical research. Her approach is grounded on collaboration with internal and external study team members to ensure clinical pharmacology objectives are appropriately assessed and the findings are clearly communicated.
Thank you, Ryan. We've heard from the rest of the speakers about the various strategies to reduce patient burden and increase enrollment. I'll be talking about leveraging clinical pharmacology to reduce patient burden. Next slide. So since the primary objective of these studies is generally to assess the effect of renal and hepatic impairment on the PK of the drug, it's really not surprising that we can use clinical pharmacology tools and considerations to guide decision-making and also potentially reduce patient burden and save time and money. This is a very broad topic, so I'll just be doing a very basic overview and limiting my discussion to reduced and adaptive study design, population PK modeling, and physiologically-based PK modeling. Next slide. So before we get into those specific tools, it's also important to note that the clinical pharmacology considerations go into the decision as to whether to have a dedicated hepatic impairment or renal impairment study in the first place. So first, looking at a hepatic impairment study, I don't have all the details from the guidance here, but just glossing over. There's definitely an overview of considerations for where a dedicated hepatic impairment study is required, as well as when a reduced study design may be appropriate or other approaches. This can include things like the extent of hepatic metabolism and elimination, as well as the therapeutic index of the drug. It can also take into consideration things such as the planned dosing regimen. So for example, single dose administration usually doesn't require a dedicated hepatic impairment study. The other thing to take into consideration is the prevalence of hepatic impairment in the intended patient population. This can influence the decision to utilize a population PK approach, because you could use data from the phase 2-3 studies in lieu of a full dedicated hepatic impairment study. And of course, it would be dependent on having enough patients enrolled in your study. Next slide. There's similar considerations for renal impairment studies based on the regulatory guidance. I won't spend a lot of time here, but I wanted to highlight a few things. First off, renal impairment can also impact protein binding, hepatic enzymes, and transporters. So renal impairment studies may be important for drugs that are primarily metabolized by the liver and demonstrate extensive protein binding, for example. Additionally, consideration to the target population is also required, because if the drug is given in patients that receive renal replacement therapies such as dialysis, then the impact on PK will need to be evaluated, as we've already heard. There can also be exceptions, of course, for drugs with high molecular weight or with extensive protein binding that are not affected by renal impairment, as these would be unlikely to be affected by the dialysis. Next slide. You've already heard about reduced design, and in some cases, this is all that's needed. For example, a drug with limited hepatic impairment, reduced study design, just taking moderate hepatic impairment compared to normal function may be sufficient. Or for a drug eliminated hepatically, a study in severe renal impairment compared to normal function may be sufficient. Additionally, reduced design is often used in combination with adaptive design or POP-PK modeling, and I'll get into a couple of these examples in the upcoming slides. Next slide. Our first example will be a reduced or adaptive design in subjects with hepatic impairment. So we would start with subjects with moderate hepatic impairment and compare those to normal function. And if you don't see any effect, then you're essentially done, and you can apply the results to subjects with mild hepatic impairment. But then if you do see an effect, you could make the decision to proceed to test subjects with mild hepatic impairment. The other alternatives are to use the reduced design in combination with the phase 2-3 study results. So you could model the results from those phase 2-3 studies if you have sufficient subjects, for example, subjects with mild hepatic impairment, and then that would be in place of having to do to test those subjects in a phase 1 study. Additionally, the guidance generally recommends contraindicating in patients with severe impairment if you've done this approach, but if you are enrolling sufficient patients in your phase 2 and 3 studies with severe impairment, you could utilize those results to determine the effect of severe hepatic impairment on the PK of your drug. Additionally, in some cases, sponsors have decided to just utilize the results from phase 2 and 3 studies and use POP-PK modeling, but oops, they get to the end and they didn't enroll enough subjects of the varying degrees of hepatic impairment. In that case, you could start with a POP-PK modeling and then add on a reduced design to supplement those results. Our next example is a reduced design for a renal impairment study, and this one is outlined in the renal impairment FDA guidance. So this would be commonly applied for drugs that are primarily eliminated hepatically, where you still do need to test the impact of renal impairment. So you would start with the worst case scenario, so subjects with severe renal impairment but not on dialysis. And then if you have no effect, again, you're essentially done. You can apply your results to those subjects with mild and moderate impairment. But then if you do have see an impact on PK, then the guidance recommends that you proceed to do a full design testing subjects with mild and moderate impairment. However, you can, again, use data from phase 2 or 3 studies and use PBPK modeling to supplement the results of the reduced design and model in those subjects with mild or moderate impairment to avoid having to do a full study. Next slide. The other new approach that's rapidly evolving is physiologically-based pharmacokinetic modeling, or PBPK modeling. This combines mathematical models of physiological, physiochemical, and biochemical processes with available PK data from whatever you have from phase 1 studies or even non-clinical studies to predict the PK and desired scenarios. So this can take into consideration all the physiological changes that are associated with renal and hepatic impairment and the impact on a drug's PK. So you're essentially building a virtual patient. This is, again, a rapidly evolving area, and while the predicted performance varies, PBPK modeling is recognized as useful for predicting exposure, particularly with respect to planning for study design and dose selection for your dedicated studies. It can also be used as a supplement for scenarios where that would be difficult to study in a clinical trial. There are a lot of recent examples where steady state PK is predicted based on single dose data, or even looking at the effect of drug interactions in patients with hepatic impairment. Next slide. So just considering all the different things we've discussed today, reduced and adaptive design are helpful in limiting the number of subjects that need to be enrolled in these studies, and they have very straightforward objectives that are easy to test, but there can still be enrollment difficulties, and it also won't look at the impact of disease in combination with renal or hepatic impairment. PBPK modeling can be a very useful tool to supplement the reduced design or to test when it's contraindicated to test in healthy subjects. Additionally, using patients means you're able to assess the impact of disease on hepatic and renal impairment. So that would be when you're using your data from phase 2 and 3 studies. But you do need to take into consideration proper design of those studies and take into consideration the prevalence of hepatic and renal impairment in your studies. You also have to be careful that you're collecting the correct data and assessing measures of hepatic and renal impairment along with your other assessments. And then finally, population PK modeling is useful for study design optimization and for predicting the impact of hepatic and renal impairment for scenarios that would be difficult to study or even contraindicated. The downside is it is complex and can be, it requires a lot of data to develop and validate. And the predictive performance can vary, particularly for the higher levels of hepatic or renal impairment. But this is an area that is rapidly growing, and as we gain more information, may become an even more powerful tool. Thanks everyone for your time today. Ryan, do we have any questions from the audience?
Well, thank you very much, Dr. Grant, and everyone for this very insightful presentation. I'd like to invite our audience to keep sending their questions or comments for this Q&A portion of the webinar. If you're attending via GoToWebinar, you can use the questions window. And for those who are watching on LinkedIn Live events, feel free to post your questions in the comments section. I've already received some questions from the audience, though, so we'll get ourselves started with those. What pharmacodynamic endpoints do you find most relevant when interpreting data from studies involving hepatic or renal impairment?
Excellent question. In some cases, PD endpoints may actually be the primary objective. For example, if concentration response data aren't available, or they can also be a secondary endpoint if there's concern about the PD response being altered. It's important to know that in some cases, PD could even be altered independent of the pharmacokinetic changes. And so both the regulatory guidances do recommend dialoguing with the agency, to discuss which PD markers should be assessed. But on a related note, on most studies, you're also commonly characterizing the relationship between measures of renal impairment and hepatic impairment on PK, either using basic linear regression or more sophisticated modeling. And you can also be looking at things like exposure response relationships. That can be important in understanding the impact of renal or hepatic impairment on the safety and efficacy of a drug. Did I hit everything? I feel like I missed something in there, but sorry.
No, that was great. Yeah, thank you very much for that answer. I've got a question here for Corey. Someone wants to know, for planning purposes, how far out should discussions be initiated with the CRO and sites?
That's the $1 million+ question. Ideally 6 months out, maybe further. It depends on study design, the N. A larger study, you might need 9 months. It's to make sure the resources are reserved, the sites are contracted well in advance, and they can start helping with protocol design as their feedback is critical.
All right, very good, thank you. Now, Dr. Alcorn, I've got a question that's come in for you. Someone's curious, why do we not want to do an eGFR on day one?
Oh, good question. So the big risk that you run is that the lab could be delayed or you don't receive it in time for day one dosing. Therefore, the patient wouldn't dose. And typically patients don't change during the screening period with their eGFR. And that's eGFR is just that. It's an estimated renal function test. And if they qualify during the screening period for the category on what you're recruiting for, that is typically what's seen in most renal protocols. And FDA clearly is approving these studies, and they don't have specific guidance as it relates to the requirement for this, as because this is an estimated GFR, not a true GFR.
All right, and another question here from someone, why should we not start AE or SAE at ICF?
Again, when the specialty population, these are people with comorbidities. In the renal population, you have diabetes and hypertension. Hepatic populations, you have alcoholism, IV drug abuse, obesity, whatever. The point being is that these are complex patients who are going to have adverse events. And having those adverse events documented and tracked may lead to data that may be useful, may not be useful, and what good is it is to be determined by the sponsor. So why add extra costs or risk to your study until the IP has been administered, at which time IP should be tracked for AEs at that time.
All right, thank you very much. Now, I'd like to move to a couple of questions for Dr. Atty. Someone's wondering, what are some of the biggest challenges when reviewing abnormal labs?
Well, it depends on your frame of reference. If you're used to normal, healthy individuals as your day-to-day clients or participants, these can be a little bit scary to deal with when you start seeing these labs that are significantly out of range. But again, the thing to remember is that this is their baselines. Obviously, it's very helpful if you have data that confirms that, and that this wasn't a one-off that just came in and it was, all of a sudden their LFTs are sky high. So that is something to consider, always having that review or the ability to review previous labs. But the good thing is that the fact that you find them, obviously, in many cases, it's a requirement that there be abnormalities in order for them to be enrolled in the first place. But the likelihood of you having to intervene is very, very low, and it's more just a matter of monitoring.
All right, very good, thank you. Now someone here is curious, how can I recruit patients from my practice to enroll in this type of trial?
Well, as I mentioned earlier, the fact that these subjects are in your practice is perhaps you're not actually aware of that, but it does pay off to do some data mining, especially from the renal side. Obviously, if you have a patient that has a significant hepatic impairment, you're probably going to know about that What you may not know is somebody who, some middle-aged, maybe 60-year-old female who has mild renal impairment or maybe even moderate renal impairment. So it's important that if you identify these subjects or you are looking for these types of patients, that you do it a little data mining, and you'd be surprised as how many would actually qualify for some of these studies.
So back to you, Corey, for a moment. The question here is curious about how many sites do you recommend for these studies?
It goes with how far out should we plan for these studies. Again, it's the size of the project, the end, budget, how fast you want the data. There are economies of scale here. So as you water down the project, so to speak, if you have more sites on there, you have less subjects per site and less matches per site. And that might not be as attractive compared to other studies. So it's a balance between having the right amount of sites that can meet the timelines and your budget, and also keep the sites happy. So I would say anywhere from 2 to 3 is on average a good number.
So Dr. Alcorn, I've got a couple other questions here for you. Someone's curious, how can we be assured that renal function in renal patients is stable?
As it relates to renal patients with stable renal function, it's clearly how you define stable renal function. And typically patients don't transition from one group to another unless there's an outlying issue, be whether diabetes or hypertension respectively. To that point, the stability could be verified with a double sampling within the 28-day screening window, but must be done at least 3 or 4 days ahead of day 1 to ensure there's not a delay in dosing. This would require a second visit and an additional cost to the sponsor for the site to get the actual sample for testing.
For the next question, someone's wondering, do we need to allow for repeat blood pressure reading during screening?
In the specialty population, we would encourage this and recommend this, as these patients do come in and they can be on the border of the exclusion for blood pressure, as these are hypertensive patients in the renal groups, as well as in the hepatic groups. They can also be on the border. They can be concerned about the white coat syndrome, where they get anxious seeing a clinician in a white coat. So you want to make sure that they feel comfortable in the environment, and you allow them to feel that they are stable and willing to give you readings that then can be recorded appropriately that meet the inclusion-exclusion criteria.
Right, okay, thank you very much. Now, Dr. Atty, I've got a question here wondering, how do you manage participants with significantly abnormal labs while they're in your unit? First
of all, as I mentioned earlier, these lab levels are frequently abnormal when they're screening, and this is, as I said before, where they live. The thing about these types of trials, generally they're very short-lived, so it's not like you're going to have to dose them every day in most cases, or actually even have quite a few blood draws other than the PK sampling. So there's not a whole lot to manage. The thing to manage is the level of discomfort that you have when you are taking care of these subjects. Typically for family practice, internal medicine, we don't take care of these by ourselves. We are usually involving the GI specialists and renal specialists. So knowing what you're starting with, knowing what you're dealing with, and making sure that the patients are safe and watched closely. There's not a whole lot to manage when it comes to these abnormalities.
Okay, thank you very much. And Dr. Grant, to wrap things up, I've got a question here for you. Someone just wondering, are POP-PK and PBPK analysis accepted by regulatory agencies?
Yeah, great question. Absolutely, they are. POP-PK modeling is baked right into the renal and hepatic impairment guidances, and so definitely proposed by the regulatory agency. So long as you are making sure you're collecting sufficient data, collecting data from the different populations of hepatic impairment, varying degrees, so that you can have a really good analysis and, all the different caveats that go along with that. There's obviously more details in those guidances. And PBPK modeling, while it's new, is becoming more accepted and being most often used in combination with other analyses. So for example, I think it's isavuconazole, the antifungal drug. There was some data in mild and moderate hepatic impairment showing some reduced clearance, but then nothing in severe hepatic impairment. They were able to use PBPK modeling to predict the impact and then have some recommendations for dose adjustment. And particularly as we're seeing it being more difficult to enroll that severe hepatic impairment group, this can be a good starting point for physicians to know what to do about dose adjustment in that population if you haven't been able to have a dedicated study in that population. So, yeah.
All right. Well, thank you very much for that answer and all of you for all of your answers today. But we have reached the end of the Q&A portion for the webinar. If we couldn't attend to your questions, know that the team at Dr. Vince Clinical Research will follow up with you after the presentation. And I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Additionally, I have shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join me in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.